Welcome to the Classics of Immunology Journal Club. We're now up to 1977 in our saga of papers, starting in 1798 with Edward Jenner. Before we get started, I just wanted to say, if you like this journal club, go online to the YouTube site, like it, click on the little bell so you get notified for more. We're getting more and more people that are following this. And then also go on, scroll down and go into my website, because you can get all of the classic papers are there for like the last 200 plus years, at least the ones that I consider to be classic. And also the social media sites are there, particularly Twitter and Instagram. So the paper from today is by Steve Gillis and Kendall Smith, myself. This is one of our very first papers coming out of Dartmouth Medical School in Hanover, New Hampshire. It's entitled The Long-Term Culture, Tumor-Specific Cytotoxic T Cells. And this was the beginning of the saga, essentially. This was the progenitor of it all. It was published in Nature in 1977. Just to give you a little background on these two authors, Steve Gillis was a graduate student. He'd just begun. He was just there a year or two in my laboratory. And I was just a young assistant professor, having joined the faculty at Dartmouth in 1974, just 3 years previously. I had been on a postdoctoral fellowship in France and just outside of Paris. And when I came back in 1974, the first thing I did was sit down and write a grant to study tumor immunology. That's what I wanted to do, because my clinical training had been in hematology and oncology, and I wanted to look for tumor-specific immunity, which is what this paper really is about. So the first grant was funded in 1975, which is 2 years before this paper came out. So in the introduction to this paper, we sort of list sort of the background of these studies. And so because the paper is about long-term T cell lines, we point out in the introduction that most human T cell lines had been able to be created from B cells, not T cells. However, in almost all instances, whether or not they were from leukemias and lymphomas or whether they were from normal B cells, these cells had been infected with Epstein-Barr virus, which was thought to and then shown to be the reason that you could establish a long-term. They were really malignant B cells as a consequence of the Epstein-Barr virus, which is a DNA herpes virus. T cell lines had not been really established except in exactly the same numbers as B cell lines in the human. And in the human, they all were from ALL, or acute lymphocytic leukemia, of the T cell variety. And the T cell lines that were available or established didn't have any immunological functions. They didn't make cytokines. They didn't kill. They didn't do anything essentially except grow. Now there were in 1974 there was a paper from Rob McDonald and Jean-Charles Serratini and Ted Brunner from Lausanne had shown that if you in the mouse you could get T cells to grow longer term than just a few 2, 3, or 4 days or a week max. And in order to do that, you had to then harvest. You could put cells into culture in a mixed lymphocyte culture, so that the responding cells in the MLC, one-way MLC, were being stimulated by allogeneic or histoincompatible lymphocytes of another mouse strain, so that you could activate a proliferative response that would peak at about 4, 5, 6 days, and then it would start to wane. If you harvested those cells, re-stimulated them with another batch of stimulator cells, which were simply either exoradiated or mitomycin C treated spleen cells, lymph node cells, then you could get a secondary response. And if you could keep on doing this, McDonald and his cohorts showed, and you could get cells to grow for 2, 3, 4 months. That was interesting. This was all based on the dogma that you needed an antigen to stimulate the cells. And that was really solid in immunology, that the lymphocytes would not become activated and respond unless you activated it via the antigen receptor. Now, of course, in the early 70s, we didn't know what the T cell antigen receptor was, what it would consisted of at the molecular level. People had identified that in B cells, there were surface antibodies, surface immunoglobulins, and those were the antigen receptors on B cells. But T cells didn't have any surface immunoglobulin, and we didn't really know what was going on in that instance. And also in the human, there was a report in 1975 that showed that if you could do the same thing that McDonald did with repetitive mixed lymphocyte cultures, you could do that in the human. And this came out of the Karolinska Institute in Sweden. Again, the cells just would grow when you re-stimulated them every week or two by harvesting the cells and then putting in more re-stimulating cells. Then in 1976, just a year before this paper that we're talking about today, it was the Morgan Rossetti Gallo paper that we talked about on the last episode of the Journal Club, where they showed that if you took normal human bone marrow, you could put the bone marrow in culture, and you could grow these cells continuously over several weeks, as long as you kept them in lymphocyte conditioned medium. And that was their abbreviation for it, what allowed these cells to grow. If you replace the conditioned medium with fresh medium, the cells would die within 24 hours. So it was the conditioned medium from PHA stimulated normal human peripheral blood lymphocytes. At the time that Morgan and Rossetti and Gallo did that, we had been working on, in the mouse, not in the human, to generate cytotoxic T cells against mouse leukemia cells. And we had a paper in press at the time that this paper came out. We had a paper in press in the JEM showing that you could get enhanced killer activity if you did two things. One is you could use allogeneic tumor cells to activate spleen cells or lymph node cells from mice, and that would then kill the stimulator leukemic cells. They would kill both allogeneic tumor cells or syngeneic tumor cells from the same that had been undergone a malignant transformation as a consequence of the same RNA tumor virus. And in this instance, we were working with Friend leukemia virus from Charlotte Friend from the Sloan Kettering Institute. So what we wondered when we heard about the Morgan paper, whether we could use this to keep our cytotoxic T cells. We could generate the cytotoxic T cells in mixed tumor lymphocyte cultures, and we could do repetitive mixed tumor lymphocyte cultures, secondary, tertiary, quaternary, just like McDonald had shown with MLCs. We did the same thing with MTLCs. So we wondered whether or not we could keep these cells growing in this lymphocyte conditioned medium stuff. Now, I thought, basically that was a long shot, because the immunological dogma was that that wouldn't work because you needed antigen. And if you didn't resupply the antigen periodically, these cells were going to ultimately peter out and die off and so forth. But we thought we'd give it a try. Because we knew that if we could get these cells to grow in this lymphocyte conditioned medium, whatever it was, that then we could generate huge quantities of killer T cells that would kill leukemia cells. And because that's what I really wanted to do as a young hematologist, tumor immunologist, I thought this was a good thing to do. So in the methods part of this paper, in order to do a secondary, primary, and then a secondary mixed tumor lymphocyte culture, we worked with C57 black 6 splenocytes that we could stimulate in vitro with a Friend leukemia virus cell line called F45 that was from the DBA2 mouse, which was a different histocompatibility locus. We put those cells into 2% fetal calf serum, and in a very special medium that was called Click's medium. Click was an investigator at the University of Minnesota who had come up with a very enriched tissue culture medium that was specialized for mouse cells, and it was clearly better than Delbecco's MEM or 16 RPMI 1640 or any other. Our cells really they loved Click's medium. So we put them in to this MTLC for 5 days at a responder to stimulator ratio of 40-1. So 1 stimulator cell to 40 responder cells. And we got cells to grow, become activated and proliferate. And then we did a secondary stimulation for only 2 days. We put those cells then into the lymphocyte conditioned medium, and then they grew. And I'll get to that in a second. The other thing about them, Maura, just like I said before, in terms of the primary, secondary MTLC, the killer T cells would kill both allogeneic stimulator cells, but they would also kill syngeneic leukemia cells. That was very, very important, essentially. The conditioned medium we used it at a 50-50 mixture. And the cell line that came out of that initial culture was called CTLL1 for cytotoxic T lymphocyte line 1. And then we also did we got a second line that we started at exactly the same time, where we had immunized the responder mice, the B6 responders, with allogeneic leukemia cells in vivo by injection into the foot pads, and then waiting a week or two, and then we would harvest the spleen cells and do a secondary and tertiary mixed tumor lymphocyte culture, and then put those cells into the conditioned medium. And the cell line that grew out of that, we called CTLL2, cytotoxic T lymphocyte line 2. Now, we made our conditioned medium. We didn't use PHA because we knew that in the mouse, the concanavalin A was a better mitogen, was a better stimulant than PHA was, which was better in the human. So we just winged it. We said, well, it's got to be better. So we made our own Con A activated T cell soup, and that became derogatorily referred to by some people in the business as just catsup Con A activated T cell soup. So that was the materials and methods. And for the results, I'm going to do some screen sharing now. First of all, before we get to the screen sharing, I have to say that both of these cell lines, we kept them growing continuously as long as every week or so, we split the cultures down and put them in fresh conditioned medium, new conditioned medium. And we had them growing for 22 weeks, essentially, just over 5 months. So let me screen share with you. So this is figure 1 from our paper, which you can see are representative growth curves of CTLL1, which is in the closed circles, and CTLL2, which is in the closed boxes. And the dotted lines indicate where we split the cultures down. And so very, very importantly, we seeded the cells at a low cell density of only 5,000 cells per ml. That turns out was critical, and that was the reason we were successful. If we seeded them higher, and usually immunologists always put their cells into culture at about 1 million to 2 million cells per ml, because they were trying to get as much thymidine incorporation as possible, and they never really looked at them after they put them into culture. But these cells were seeded at a very low cell density. And so you can see that over the course of a week here, this is total viable cells per culture times 10 to the third. And over a week, they grow up almost 100-fold increase from 5,000 to 500,000. We then split them down, and they will just keep growing like this forever and ever if you do things right. So that was figure 1. And then what we did, so we were happy that we could get these cells to grow because they were sort of antigen selected. And we knew when we started these cultures that they were cytotoxic cells. So we tested them periodically over time. And this is this figure 2, or that's figure 3. What happened to figure 2 here? CTLL1 curves on the left in figure A, and CTLL2 in figure B. And what we've got here on the ordinate is the percent cytotoxicity, and on the axis is the effector to target cell ratio, going from a low effector to target cell ratio up to 100 100 to 1. And then we tested these cells at 3 weeks, which is in the closed circles, and at 17 weeks, and at 33 weeks. 17, no, let me see, no, sorry, 10 weeks, and then 17 weeks. And the curves march to the left because we were able to get the same amount of killing. Let's say if we looked at 30%, which was our definition, here we need 10 to 1, and down here we need like 2 to 1, and down here we need almost 1 to 1 would kill those cells equivalently. Then, and so this was CTLL1, this is CTLL2, same thing happened. The target cells in figure 2 were all allogeneic target cells, the stimulating tumor cells. Here, we've used syngeneic friend virus induced leukemia to the B6 mouse so that they're FBL3 cells. And you can see the same general phenomenon happens. This is CTLL1, this is CTLL2. This is at 3 weeks and 17 weeks, or 10 weeks and 17 weeks. And this is the effector to target cell ratio. And these open circles or open symbols on the axis here are normal target cells. We used either lymph node cells and or Con blasts, spleen cells. And you can see that that was negative. And that's a very, very important point because where we were coming from was we weren't interested in just allogeneic cell killing. We wanted to show syngeneic tumor-specific killing, and that's what we were able to see there. We also tested these tumor cells for evidence that they might be making virus, because then you could say, well, it's not a tumor-specific antigen; it's a virus-specific antigen. And we showed that the tumor cells were non-virus producers. Presumably, then the antigen being recognized was a tumor antigen rather than a viral antigen. The last figure in this paper are cytospin preparations of the cultured T cells. And the first, and this is a Wright-Giemsa stain, which is the normal stain that the hematologists use when they're staining blood cells. And you can see that these are mononuclear cells on the one hand, and you can see metaphase plates here and here. But the other thing that's very striking about these cultures is that these cells are chock full of granules, and so that they look empty because the alcohol fixation sequence that is used washed out whatever were in those granules. And when we first saw this, I thought to myself, well, these aren't T cells. These probably are macrophages. The good news was that wasn't true. We looked for T cell markers, and so that we could show that they were theta positive. We also looked for, we had special stains that we could use for all of the myeloid cells, myeloperoxidase and those sorts of things, and those were all negative. So we could say by all the things that we did that these cells were T cells, and we knew they were cytotoxic T cells, and we went on from there and got electron micrographs of these cells, and where we found that these granules in the cytoplasm were electron dense granules, and subsequently, and it took maybe a decade, but all others then showed that these granules were cytotoxic. If you isolated the granules from the cells, broke open the cells, and isolated the granules would kill target cells directly. And within those granules, what was found was the protein perforin, which is what pokes the hole in the membrane. And then there were also proteolytic enzymes that were labeled granzymes because they came from the granules that were cytotoxic in the whole thing. So that's all the screen sharing. That's all for the data in the discussion of this paper. we make several points. You know, this is, again, this is the Nature paper, right? It's only 2 pages essentially. So you have to be very concise in your, you can't just go on and on and on. So we basically say, all right, number one, because of these cells seem to be able to grow in vitro continuously and constantly, that means there must be something in, I say in vitro, there must be something in vivo that regulates the lymphocyte growth and turns it off eventually. Seemed like a logical proposition. Secondly, the fact that you can grow these cells long-term, continuously and constantly, that means that we can probably use these cells to make an assay for the whatever it is in these conditioned media, the so-called growth factor. And we ought to be able to then purify that growth factor and to characterize the growth factor. That would be good. And thirdly, because of the fact that you can grow these cells up, you can get unlimited numbers of them. That means you ought to be able to use these cells for immunotherapy. You ought to be able to then take a mouse with leukemia and inject the mouse with these cytotoxic T cells, and that should be beneficial. So we were really skewed on towards tumor immunology in this paper, which was sort of a side field at that time. No, people didn't look kindly on the whole idea that the immune response might be therapeutic for cancer or tumor cells. And the reason I made a big deal out of the fact that these were non-viral producing cells is that human cells, human leukemia cells, nobody had been able to find a virus being produced by the leukemia cells themselves. So therefore, the experiments were meant to mirror what was really the human situation. And that was very, very important. And also the very fact that we were able to get antigen-specific T cells to grow, so that they're differentiated and they're also proliferating. It wasn't just any old antigen. This was a TSTA, a tumor-specific transplantation antigen kind of thing. So that was it from the standpoint of the points that we tried to make in this paper. And we sent this paper off, and I was quite confident that this was going to fly with not any problem wherever we sent it to. So we sent it off to Nature, and we got it back within a week. And I think I've said this before, maybe one of the other or several videos that I've made. We got it back rejected, and it had never been sent for review. They said they weren't interested in it. And that made me, first I was shocked. Couldn't believe it. And then I got angry. So I sat down and I wrote a letter to the editors, and I explained to them in 1 page. I didn't think it would be a good idea to go on and on and on. They probably wouldn't even read it. They just put it in the circular file. I tried to explain to them, or I did explain to them, why this was an extremely important paper. This had never been done before. Nobody had been able to grow T cells and keep them growing long-term and have them being functional, number one. Number two, against a tumor-specific antigen. And this was going to be a revolution for T cells. So they sent it, and I said, you should send it out for review. So they sent it out for review, and the reviewers agreed with me, which is a good thing. And it was published on Bastille Day, July 14th in 1977. And I always thought that was right immediately. I said, this is a good omen. This is going to be our ticket out of here, essentially. And that was true, because it really was the beginning of a revolution for T cells, because now you could get large quantities of these things. We could approach the growth factor, find out what it was, and so forth and so on. And we're going to be reviewing those papers as time goes on in this journal club. The other thing about this is that, so it came out in Nature in July 14th, 1977. And the next spring, I went to the spring FASEB meetings, Federation of Experimental Biology, where the immunologists were the the card-carrying immunologists would all congregate in the spring. And I expected, a lot of people to be excited about this, talking about this and so forth. And there was zero. Nobody had heard about it. Nobody would pay attention to it. And it was sort of deflating. But, and that was because the immunologists themselves were essentially steeped in the dogma that nothing could make cells, T cells, lymphocytes proliferate except the antigen. And of course, that's true. Antigen initiates the process, but the molecular mechanism whereby it takes place, the growth factor, is the thing that makes the cells grow. And the fact that the card-carrying immunologists didn't realize that this was a very important phenomenon gave us time. So we were up at Dartmouth. Dartmouth was not known for immunology. There were no other immunologists at Dartmouth. And so we just worked away, did our deal, and for at least 5 years and maybe more. And we were way, way, way ahead of everybody else. And that was a good thing. So it was good that nobody perked up and said, hello, this might be important. So that's the end of this journal club. And don't forget to like it on YouTube and click on the bell and check out the social media sites. And wait for next week. After this one, we'll have another ringer for you.